Welcome to today's SNOMED CT research webinar. I'm Susie Roy, the host of these research webinars for SNOMED International. At SNOMED International, I'm the collaboration specialist as well as customer service representative for the Americas and research engagement lead. Always a joy to have these research webinars, to see our research community come out and support our community of researchers. So as I mentioned, there are just a couple of housekeeping items that I wanted to mention. I see a number of our returning research webinar guests, past presenters. Welcome to all of you. And I also see a number of new people joining. So welcome everyone. Today, you'll see that everyone is muted. However, if you have any questions as we go along, go ahead and pop those into the Q&A box in your Zoom webinar screen, and you will be able to ask questions at the end of the webinar via that Q&A box. Sylvia will be glad to answer your questions at that time. In addition to our research webinar, we also have a clinical webinar and implementation webinars. All of those for past presentations as well as upcoming presentations can be found on our website, snomed.org/web-series. I highly recommend everyone to go and check those out. If you wanted to see those past presentations, we also have a SNOMED International YouTube channel. I think that all of our past research webinars are indicated or listed under a playlist entitled SNOMED Research Webinars, but we also have other great presentations from past expos as well as keynote presentations and much more. So I highly encourage people to go ahead to our SNOMED International YouTube channel and like and subscribe, of course. And if you enjoy today's webinar or just want to be informed of upcoming research related news to SNOMED CT, you can join our listserv. That is, to join that, you just have to email me. I'm Susie again, sro@snomed.org, and I will get you on that list, and you'll be notified of not only research webinars, but also upcoming research related events, as well as other sorts of SNOMED CT research related news, such as new and current research that's happening in our community. One of those things that I frequently update everyone on is our SNOMED CT Expo. So our SNOMED CT Expo is our annual conference that happens sometime in October. And it's basically a great way to meet and experience the world of SNOMED CT. We have everyone from government member representatives to research community to clinical experts who want to share their SNOMED CT expertise. So that's happening this year in Atlanta, Georgia, the United States, from October 26th to the 27th. And I highly encourage those who are interested to attend. You can scan the QR code that's on your screen or go to our website to register for that awesome event. During that event, we have multiple streams. Some of those streams include the demonstrating implementation excellence, enhancing clinical practice and outcomes, and the terminology leading practice. And also starting last year, and we're continuing this year because it was a huge success, advances in research and innovation. And of course, I always have to thank the research community for coming out and supporting that. So again, it's a great way to just explore various sessions, tutorials, poster presentations from our community of practice and research. New this year is a pre-conference event. So for those who are well aware, but for those who are not, the beginning of the week starts our business meetings. So that's all of our governance meetings. And then the end of the week ends with our Grand SNOMED CT Expo. This year, we also have on that Wednesday, so in between the two events, a number of symposiums and special events. So those who are interested, we have SNOMED CT and LOINC session going on in the morning. We also have a SNOMED CT implementation support management and Simplex toolkit tutorial and demonstration going on, and a clinical seminar. I'm very excited because new this year, we have the SNOMED CT AI Symposium, and it should be a great event. So if you have any questions, go ahead and ping me, but I hope everyone's able to join one of these awesome events. All right, so I'm almost done talking because I know you're all here for the grand event. So Sylvia van Laar graduated in 2018 with her PharmD. And then currently she's getting her PhD at the Leiden University Medical Center, the Netherlands. She'll defend this thesis later this year. And currently she's working as a policy advisor on semantic unification at the Dutch Ministry of Health, Welfare and Sport. Today, Sylvia will be presenting text mining real-world data to evaluate systemic anti-cancer therapy. Sylvia, thank you so much for joining, and please take the screen.
Thank you, Susie, for this introduction. And that's very nice. I'm going to share my screen so you can all see what I'm seeing. Perfect. Yes, thank you. As Susie said, I'm now currently working for the Ministry of Health, but before I did research for my PhD, and the title of that will be Text Mining Real-World Data to Evaluate Systemic Anti-Cancer Therapy. And last April, I was on the SNOMED meet in London, and that's where I got talking to Susie, and she said, like, hey, come by and present this if you want to. So I'm very happy with this invitation, and I will tell you my story of what I did in my research. And first, well, that's already set for just a detail, but I did all this research as a PhD student, so that's not linked to the Ministry of Health, of course, and the sources are mentioned on the slides, and some screenshots which are in there might be outdated, but those were the screenshots from when I used the programs. So just wanted to have that clarified. So first to start, this is what I want to discuss with you and talk to you about, of course, first an introduction into the topic. And then I will explain a bit more about the text mining tool I used and how it worked and what I did with it. And then I will take you with all my through all my study outcomes. And in the end, I hope there will still be some time for discussion, and I also have some discussion points. So I'm curious what you will think of those. So first, my start. As already mentioned, I have a PharmD, so I studied pharmacy, and one thing I was always like remarked by is how you have this very long list of well, very nice new cancer therapies, and they're registered, and there's this balance between you want them on the market as soon as possible because there's a high need of those treatments, but also you want to be sure they are effective and safe. And this graph shows very nicely, I hope you can see it in general, how close the points between the publication of the registrations are in green and the approval of the EMA or the FDA was in blue or orange. And sometimes even in cases, the publication of the data was after the market approval. Well, what that means in general is that even though there is some information and in baseline they are approved, we still do not know everything about those treatments, and in clinical practice there's a lot of small questions to be answered still. And this graph also shows it very nicely. In the beginning, of course, you have those trials where data is generated to have this approval, but actually after the market access, you get the large generation of data and also the data you can use to answer a lot of other questions. So why do we actually want those other types of data? Of course, the trials are very good due to their randomization. So what they answer is, does this treatment work or not? And is the effect due to the treatment? But of course, there's often a limitation to what patients can participate, and there might be other developments after registration. And with the data from clinical practice, you can answer like, does it also work, for example, for a patient with poor kidney function, or there are more side effects, very rare side effects you only see in that case, and there might be a lot of things happening differently in clinical practice than in the trial. And especially with those treatments, those cancer treatments with the limited data for registration, you want to know you want actually to know more about that, or at least that's what I wanted. So that's why I continued with this research or started it. So this is an overview. It's not everything, of course, but this is a very general overview of what kind of sources of real-world data do we actually have. And of course, in the beginning, you have the case report forms used in the trials, which means in basic, you can collect all the data you want. It's done by staff, measurement, lab values, everything. Patients are there. But of course, this is manual labor. It's very timely and costly, and you cannot do that for everything. So on the other side of the line, you have a lot of other types of data. First, the electronic health records. I think for all of you, that's a known source, and it's nice that there's a longitudinal rich data generated through routine care, independent of the disease or treatment. It's a different type of health record you want to access, for example, in a hospital or in a GP's record. But there is some line for one patient. And then you have the claims data. Well, this is my perspective of the Dutch perspective, so this might be different in your country. But there's more general overview of, for example, treatments or surgeries or stuff on a higher level, but only if there's like need for a special claim. So on one patient, you can get a bigger overview of what has happened to it, but there might be a sense of sensitivity that might be not always very high. Then you have the patient-reported outcomes. These are the questionnaires who are set out actively to patients, and it's very nice, these types of information, because this is actually the story from the patient's view. You can use those for the quality of life of a patient, but a lot of experiences you can collect with this. Also, there's a time intensive thing. The patient has to get it's like it has to have something extra for the patient. Everything has its limitations, of course. Then you have the registries. This is more a collection of data, but here there might be data collected from the records or from other sources, which is very nice because they're regional or national large structures with on big questions or big diseases. One of the downsides is since this data needs to be collected in the registry, there might be a lag of a few years, and not all registries are based in the same structure still. And then we have upcoming sources of rural data, as social media data and wearable devices. I have my watch also over here, but also your phone. But you can also get, there's a lot of limitations to those as well. Social media data, people might complain or explain to other patients like, hey, I experienced this, did you also experience that? But you're missing out on some demographics, for example. And you can also imagine not everyone is using this data. And on the wearable devices, you have the same thing. Not everyone might be using them. And also the quality might be different. Well, this is it's nice to know that we all have these sources of rural data, but what are we going to use to actually answer these questions? Related to oncology, we want to know a lot of things. Of course, we want to know the effectiveness. Does this actually do something for a patient? But also safety. Are there any side effects or in higher occurrence occurring? And so you have more of those topics. The reimbursement, is it actually worth it? Do we need to change this? Clinical guidance, this goes both ways. How do we need to set the guidelines? Do we need to change them based on the data? But also you can use the real data to educate your patients as a clinician. And the last thing is active monitoring. You can analyze treatment patterns. Are the patterns according to what we expected? Is there some inappropriate use or ineffective use? Well, you can do a lot. And this all sounds, of course, very nice. But then why do we still need this text mining method? Well, I hope everyone's still here because this is like the second point I wanted to discuss with you. And this is big why. Literature says around 80% of information is actually still stored in unstructured text. And this also, if you think of SNOMED, that's very structured. It's very, well, I'm very happy to hear about it every time. Like, oh, if everything was SNOMED, that would be great. But the practice is that we are still not there yet, especially when I started 5 years ago. I think something went wrong. I will share
what
my
happened
screen
there?
again.
Oh,
I don't know.
Weird. Let me see if I can get it started again.
I think I'm up and running again.
Yes.
Good.
That was
You
odd.
just continue. I see a thumbs up. Great.
Okay, yes. All right. Thank you. Sorry about that, everyone.
Oh, no problem. It keeps me sharp. Well, the standard method of collecting this data was and still is just manually opening the digital files, reading and typing it over. And as you can understand, this is very time consuming and it's impossible to do for everything. So that's what motivated me to do research in what other possibilities might be there. So this is the one time I'm gonna mention the name actually. When I started, there was this startup, it's called CityQ. They had this idea of this program which you could run locally in your hospital and could use to do rule-based text mining. So make search queries, and I will explain later on how that actually works. But to like systematically search through your health records instead of manually opening it up. And so it's locally running, so the data doesn't go anywhere, and also has a nice extra that all the data you see is pseudonymized. So I didn't see any names or any addresses or stuff like that. It was all implemented. And that's what I showed on the graph on the right side, but that might be a bit impossible to read, or I find always a bit difficult. But what does this actually mean, this tool? Well, we had, I could use two types of parts for those queries. I could use structured data. I could, for example, registered allergies or medication requests or measurements, including lab values. Those are very structured data, but also could use literal words or a combination of words. And that's more like you can really go into those free texts. And still, it's nice to know that that's a possibility, but you need to think of, but how are we going to use it then? So this is our approach for if we wanted to have data from medical notes or discharge letters, etc. Those are all unstructured data. And we did data extraction through a keyword search. So we used specific terms, and we could look into, I will show that in a minute. We could actually see the sentences or parts of text where the words popped up, and you could see, hey, does it actually mean what I was looking for? So I did for everything and decided we need to do a manual confirmation. And the extra was that for the structured data, we found out this was stable. It was actually what we wanted. So we can immediately put this into the table. So what does it? I'm going to go to the next one first. So what does it look like if it goes directly into a table? You can see here length, weight, kidney function, etc. And these are like set to like, I want to have the last kidney function before start. And it just goes straight into the table. Very nice. And also you can see over here, I wanted to have like, is a receptor, does a tumor has this receptor, yes or no, so positive or negative. And I looked for terms indicating it might be positive or negative. But as you can also see over here, I know the negative, even though it said positive, because if you, as a person, still reads the context, you can know like, hey, it actually says like it's positive and less than a very small number. So you could put it into negative. So that's how we used it. And the system we actually use for it is we first use it to find patients, and then we selected those patients and then collected the data. So we constructed rule-based queries. For example, over here, if we could see for pyrexia or fever, we added a lot of words or terms and synonyms and also typos. So if any of those happened in a certain time frame, we would see it. But also we could add like, hey, fever is also above 38 degrees Celsius. So let me show those records in case it happens. If it doesn't happen, I don't need to see anything. And same for if there's explicitly no pyrexia, no fever, I also want to see it because I can put them in the different category. And with this, I could like filter everything out and I could check like, hey, in this context, it's actually pyrexia. In this one, it's not. And the last one, it's also not. So I could change it in case needed. And I could do this to select my patients and next to select the data I wanted. I showed you this already. And now you might think, well, sounds nice in theory, but does it actually work? And that's also what we thought. So we did a validation study, of course. I put in the titles where I published it. So if you want to see it, read it, you can go over there. We did a study in patients with renal cell carcinoma because this was we had a relatively big population in our hospital, and also this was a population with several different treatments and some interesting questions at. And we thought, okay, what would you actually want to collect in general? So some patient characteristics, some disease-related characteristics, some comorbidities, and also some adverse events, just to see roughly, does it work, does it not work, do we need to continue, or is it just not? So, and also we collected some outcomes. And what we did, we collected the data with the rule-based queries we constructed and did a manual data collection as well, and then compared those. Did those match or not? And we calculated the precision, recall, and F1 score, which is a combination. And next to that, since this is a question based on efficiency, we also measure the overall time necessary to verify and collect the data. So what did we find? We could include 99 patients, and those patients in total had 175 treatments. And this is the list. This is the list of those characteristics we want to check. Hey, can we do this actually or not? And a score of 100 is perfect, and 0 would be complete miss. And in general, you can see there are pretty high scores, and some are a bit lower. For example, adverse events are sometimes a bit more difficult. But also, for example, the case of prior nephrectomy, this was something we needed to collect from the free text and was actually pretty high. But next to the fact if something did occur or not, we wanted to make sure we could actually measure the effect of the treatment. And in this case, that's overall survival, progression-free survival. So we included the time and, well, compared those graphs. And from this, we concluded, well, they're not perfect, but they're very close to each other. They're not significantly different. And especially overall survival is almost the same. So in general, we said we could, with both methods, include the same population. In general, there's accurate data. Some need some improvement, some rule-based queries, but it's also 7 times faster this way. And for your, this is not in the slide, but it took me to collect the manual data. It's around 1.5 hours per patient. So you can calculate how many time I spent on that already. So next, we had this nice set of queries, of course. And we thought we can also, we can use this data to actually answer some questions. So this is the same population of renal cell carcinoma patients, but why did we want to use this data to answer some questions? Once in 2006, the first treatments were registered, comparison to the big graph I showed in the beginning. And as you can see, since then until 2021, a lot of new treatments and treatment combinations have been registered, and ongoing, there's even more coming or already registered. And you can imagine, if you are a physician, you need to explain to your patients what treatment you're going to give, why. You also need to make a choice, and that might be very difficult if you have the luxury of so many treatments in comparison to before 2006. What we did, we used these validated queries to investigate treatment patterns and also the efficacy, not only in our hospital, but we also exported the queries to another hospital in The Hague close to us. And with this, we could include 138 patients, and we found the following thing. I always call this a bit of a spaghetti, but with the data, we could show like there's a lot of treatments giving in first line, there's 3, but then you see in continuing around 60 to 70% goes to a second line, and same for third line, and some were still on treatment. Which was, well, it's good to see that patients can get multiple treatments actually. And you can also see on the right side that from starting on 2019, new treatments as nivolumab, since it was registered, are also given to patients. 2020 is low since the year wasn't finished, so not all the potential is used from that year. But I found it interesting because you can immediately see the change in when a new treatment starts. And this was the most interesting outcome we had from this data. Those graphs, I'm not sure if everyone knows how to read it, but in the beginning of a graph, everyone is still not has an event, so did not have progression, so growth of the tumor, or overall survival is they did not die yet. And in case of an event, the graph goes down. And at 50, there's like a median, so you can say like, okay, the median is around 12 here. What did we see? The progression-free survival of those 2 treatments, sunitinib and pazopanib, they were comparable to the trials, but the overall survival, interestingly, for pazopanib was longer. And we expected actually, or the only explanation we had was that all those patients in purple had the chance to have more follow-up treatments. So with this, we could conclude that it's actually useful to give those patients those treatments. It's still limited numbers, of course, but we were happy that a tool like this can help with like, hey, what's going on in practice, even though the treatment regimens can change, is it still up to date? And also our queries could be implemented in another hospital, which is very nice. Even though I have to remark that the physicians working over there also worked in my hospital or were trained in my hospital. So they're trained in the same way of registering data, which was very useful, because otherwise we might not have the same results in success. So this is the first thing we could do with this text mining tool. And the second thing is very comparable, but still a bit different, because one of the physicians said like, hey, nice that you can use this for renal cell carcinoma, but I also have a population of patients with hepatocellular carcinoma, so liver cancer. And I also want to know something about their treatment lines, how they're doing. And this is still unpublished, but I can share it with you. Okay, this is a big graph. I know. What the most important stuff over here is that sorafenib is registered in 2007, and you can see a very big gap in years until 2017. Patients for a long time only had this treatment. In the trials, there were a lot of eligibility criteria. So if you did not, for example, have child group class A, you could not participate. And this resulted all in a median overall survival extra, so the placebo versus the treatment, in an extra of 2 or 3 months, so not much extra. If you keep this in mind, you can see there's a lot of criteria patients needed to have to actually be part of the trial. So we imagine there might be patients in our population, in our hospital, not meeting those criteria. So what we did, again, we included the patients with hepatocellular carcinoma with text mining. We could select those treatments based on their structured data, of course, and we had manual validation, as I explained. And we could use the queries from before. Also, I could in program just copy them and adjust them. And we added those factors because we wanted to see, hey, does this influence our population, of course. here you can see the effect of the manual validation. We could exclude some patients because of different diagnoses, or they didn't actually start the treatment, or outside of the period. And again, a treatment pattern we could make. Just as we expected, since a lot of patients only had sorafenib, those were the most of our group, but some had the possibility to change. Since this is the biggest group, we only continued with our research with this group. But we could again calculate those survival lines, and we could conclude that those were comparable. But one thing was not comparable, and those are those risk factors we could collect. So there's a lot you can see. This is the most important one. This is like the summary. Out of the 83 patients, 61 did not, for one or more criteria, did not meet those criteria. So 70% at least is not represented by the trial. It's relatively positive news that the results are the same, but this like remarks why we actually need this data. And we could perform this. So we were very happy with this result. And I already quoted what you might have been thinking. Nice to know that we can do this, patterns and effectiveness, but what about the safety? So what about the adverse events? They might be a bit more difficult. So of course they dive into that as well. I see a thumbs up, so thank you. I'm not sure who's sending them, but that's very nice to see. So after 2 types of cancers, we went to another indication, which is melanoma treatments. And the interesting thing about melanoma treatments at 2018, 3 treatments were extended their indication to adjuvant treatment, which means a patient has a tumor, it's removed surgically, and it might be gone already, but to be sure, you give an extra treatment to be extra sure it's not going to come back. That's the idea. So you can also imagine in a case like that, you don't want your treatment to actually only do harm instead of good. So it's very important that those treatments are safe and tolerable and do not add harm, or at least in a way it's like relatable to the chance of actual cancer. Of course, it's very different field. So we included patients with melanoma, of course, again in our center in a treatment period. And in this graph, I've a bit more explained how we actually do that. So for all treatments, we have the administrative code, in this case for melanoma. And some of you might know this, but it's linked to the ICD-10 codes. But only melanoma, of course, is not specific enough if you only want to have the adjuvant treatment group. So we looked for text for adjuvant or stage 3 or synonyms of this to make sure, do we have the actual population with adjuvant melanoma? And then per treatment, we looked for prescription and for validation in the free text that they actually had this prescription at least once. So in this way, we could include them. And over here, you can see when we did the validation of those boxes, we still needed to exclude, this includes the first step, 90 patients because they were palliative treatments, palliative patients, and some switched. So this indicates also the need to add those unstructured texts still to find your exact group. Again, what did we do with the data? Well, we could, the nice thing of this treatment regimen, it's a maximum of 1 year. So we knew between a period where to look for answers actually. So we looked to see if a treatment was very well tolerated, if it was tolerated during that year at the end of the treatment, and we adjusted the system like that so we could see the texts in the health records around that time. So this is still like a bit of manual work, as in I still needed to read it, but you immediately get the text you want to have with the explanation. And then you might ask, what are these questions or what are these colors? Green is patients finish their treatment without any interruption or reason to stop. The purples, light purple, were still on treatment. The yellow patients had recurrence of their cancer, unfortunately, and the dark blue had to stop due to toxicity. And from this, we concluded, even though it's very small groups, as you can see, only 20 patients, and these are bigger, but that's the deviation between the reasons to stop was different, and then we checked this with significancy. And we concluded, therefore, that this treatment, dabrafenib plus trametinib, was less tolerable. And that's also what's over here. But that's not everything, of course. The nice thing of those trials is that you have an indication of what adverse events can we expect. So we looked in structured and unstructured data for the 10 most common adverse events from the trials. And I only have summoned here the most interesting ones. For example, fatigue. Almost all patients, at least once there was mention of fatigue in their health records. As you can see, around 80% of the patients. More interesting, pyrexia, there it is again, in 65% of the dabrafenib plus trametinib group. And in the groups of nivolumab and pembrolizumab, we saw some immune-related adverse events. And what we concluded, this is not the conclusion of this yet, even though this is a very high number, this is more into the treatments. Pyrexia, so fever, just goes away if you stop the treatment, but immune-related adverse events might be for the rest of their lives. Of course, this was already known before we started the research, but it's still, how do you say that, good? Amazes to positive, but that's like remarkable to me. That's still like 12 to 14% needs to take extra medication because they got this treatment. But the more important, if you go to like the methods, of course, it's possible to extract those adverse events, but it's still very much based on the quality of the registration of those data. If there's a lot of variability in the words used or the locations where it's written, or it's once, for example, temperature is only once mentioned in structured text or not even, it will be difficult anytime. But that doesn't differ from manual checking compared to this method. So I hope you're all still here. If you see all the studies I already showed you, you might see a pattern in the data we needed. And there's a certain timeline where you can see, of course, there's a start and a stop of a treatment. And I use those as a basis to relate the other searches for. So the first search we did, in all studies, there was a need of patient characteristics, and those are always before a treatment starts. And you can combine this with a structured search or unstructured search, but that's always a given. You need some patient characteristics. And then you always have, in this case of cancer research, there's always an outcome mostly based on survival, and that's mostly found around the time of the treatment stop, because progression or recurrence or even overall survival is mostly a reason to stop the treatment. And then we have the fourth occurrence, that's adverse events, and of course they can only occur after the treatment starts, but can continue after treatment stops. So based with this pattern, we can, investigate almost everything if you just understand the disease and the treatment and the specific patient characteristics. And that was, for me, a conclusion it was nice to have. And then you might think there was a long list of things we needed to do or we wanted the real data for. I'm checking on the time. I have two more short examples. I will give them and I'll try to be done in 4 minutes. So we have 15 minutes of discussion time. What we also did, based with the structure, you can do a lot. So can we also evaluate if a certain how do you say this? If a certain guideline is followed actually good way. So in this case, it's breast cancer, it's another disease, and this is chemotherapy. That's not the important point. It was, do patients with a high risk, so a patient with a high risk of neutropenia, febrile neutropenia, get a prophylactic treatment? And there's the case of a high, no, above 20%, and there's an interesting group of an intermediate risk. And here you see again, you have those patient characteristics you need, you have an outcome, a bit different, but we can just use the methods we have developed to answer this question. Are those treatments given? So we included a lot of breast cancer patients, 119, 77 had a high risk treatment, so in the above 20% group. This is related to the treatment, so we could just base that on the treatment they were giving, and 130 in the intermediate group. We collected all those variables as risk factors, and we wanted to know, is the prophylactic given, and did they develop the neutropenia or febrile neutropenia? And this is what we could show. So in the high-risk group, almost everyone gets it, but if it's not, it will result in febrile neutropenia mostly. And in the intermediate group, this was not what we expected, but almost none of the patients received it. And if you look at the risk factors, this is a big graph. You don't need to read it all. First, you can see we can find those risk factors in general, but still not a lot of patients receive the treatment. As you can see, for example, over here. 65 of the patients had a certain neutrophil count but didn't receive. What we concluded out of this is, next to the clinical implications, with this method, it's also possible to evaluate guidelines. You can also implement this, for example, once in a year to evaluate, hey, did the care improve in our hospital, or what's the basis? And then one last example. We all have known that COVID came around at a certain time, and during COVID, it was sometimes we didn't know what we were doing, roughly said. But it's very nice to have some data to support our reasoning. So the question came around, hey, we have this treatment now, remdesivir, and it's not tested on patients with a poor kidney or liver function, and so they're excluded from the trials again, but we are giving it to them anyways because we need to do something. Can we maybe extract those data very quickly, please, to give an indication of that doesn't do the patients harm? And I did this in 3 weeks. That shows maybe how fast we can do this. I made graphs, and as you can see, the red lines in case of liver functions were the patients who had a bad liver function at start, but they go down, which is good news. And in kidney function, you want the kidney function to be the same or go up. But the most important thing is you don't see a lot of kidney function delay below 20. So we didn't see any change, no difference between kidney or liver function at all. So we could conclude it doesn't need to be a contraindication. It's not a treatment for COVID anymore, but it is an example of why we actually need one of those methods. My take-home messages in general is it's a very valuable source, the health records, but it's limited to how the data is registered, of course, and some more uniformity is very nice and would be helpful. And this method is faster, but it still needs some manual validation. The query building is still manual, and it would be very nice if you could do this with, for example, AI, but I'm very aware that you need big data sets for this, and these are all small indications. And my dream, of course, would be that it would be an automatic collection of data with the proper privacy, of course, to registries, so people could analyze this, and that the access to those, to the records for other research questions would be better. That's everything I wanted to tell you, and I hope there's any questions.
Thank you so much, Sylvia. Yeah, thank you so much. No, this presentation was absolutely excellent. You provided extensive research evidence for the use of data analytics for both patient care and for decision support. So this was really exciting. And in particular, just the importance of just rule-based queries. And I'm actually interested to learn how SNOMED CT helps to facilitate these rule-based queries, but I think a number of other people are as well. So I will let others also write their questions in the Q&A box. So if you all notice, I'm kind of stalling so you can get your questions in there. While I'm stalling, I will also remind everyone or inform those who are new here that the presentation and also the slides will be made available. Usually I post those by the end of the week or into next week. Anyone who has registered, I will email you the direct link to these slides and to the presentation. And if you wanted to be notified of future events, any slides and presentations, even if you don't register, I always email the research reference group. So to join that, you just have to email me sro@snomed.org and I will get you on that list. So going to the questions that are currently there, and a couple of them align with some of the questions that I had. First one, thanks for this great presentation and the possibilities on text mining. In which way did your research experiment with semantic medical language like SNOMED CT? Did you evaluate it? It was more beneficial that the proprietary data you extracted in your work works out for the clinical system?
Yeah, that's the only downside. I'm aware that there's not a lot of SNOMED in actually our health records at the moment, so I couldn't use it very actively. For example, the codes I said, those are linked to the ICD-10. But it would be very nice if there's more registered, like on a uniform basis. But that's also the how do you say that? The balance I want to explain, since a lot of still in those free texts is not even structured. It's like far from SNOMED, and it would be very nice if we could reach it. And I know some examples, of course, where they used AI to link free text to SNOMED, but this is steps before SNOMED, and it would be nice if we could do that. But this is not there yet.
We've had a number of past presentations, Dr. Hohenau and Dr. Scheck, who have utilized some various tools where they were able to get unstructured data, metafied, I'm going to say. And it'd be awesome to link you guys to see, because you have kind of the next level once you have those data and what you can actually do with the those data that have been finally, SNOMEdified. So that would be really cool to link you guys up.
Yeah.
The next question comes from Kai. Thank you for sharing your research and methods. Was SNOMED CT useful in your research? Were there any parts of the patient data coded in SNOMED or mapped to SNOMED? Did you use any SNOMED queries to aggregate data or make patient selections? So.
It's actually the same. And also the most interesting thing is I was not even during this research, I was not very aware of the existence of SNOMED. So you can research health records and standardization and hear only once or twice about SNOMED. And now I'm like, if I only would have known this existed, I would have done it way differently. But you can never go back in time, unfortunately.
It's always as we get wiser as we go on. So
yes, but I think there's a lot of potential, and especially the combination if you use SNOMED with queries and just enrich your data. And also that's a way I think how to unify more of those health records by use of SNOMED.
I know you work full-time with the ministry, and you're getting ready to defend your thesis at the end of the year. So just to add more on there, are you looking at extending any of this research work with maybe SNOMED encoded data? Or is that something maybe in the future after you do?
No, I won't be extending it, but I do, of course, still know my colleagues at the university. So I can always give them ideas and inspire them. So I think that's my role for now related to the research, to just share my knowledge. And
you will be mentoring the next.
Yes.
Next question. Thank you for a valuable presentation. Maybe you addressed this earlier in your presentation, but how how do you think about power calc with RWE, real world evidence?
I'm not sure what you mean by power calc. Do you know?
I'm not quite sure on PowerCalc. Constantina, if you're able to type into the chat box, or I can unmute you so you can provide a little bit more context around that. While you, oh, here we go. Let me see if I can unmute. Okay.
Can you hear me?
Yes.
Yes.
Hi, welcome.
Hi. I was just curious when you were showing your comparisons between the different medications in one of the trials that had more than one medication. Were you concerned about whether you had enough patients to demonstrate significant differences between medications, or
Yeah.
you simply have what you have and that's how life goes, I guess?
Yes, I think it's both. For my research, the main purpose was to investigate if this method was solid and could be used. Of course, all those results are not powered enough to say anything about, in general, the
Okay.
populations. So I'm very aware that's also in all the discussions. It's not powered for that, but it gives an indication. At least, but no. So yes, thank you for your question. I hope that
Terrific.
answers.
Yes, I understand those challenges, which is why I was asking. We're facing some of similar issues in our research, and so thanks.
Yeah, and I think it's also, of course, you can always wait until you have enough patients, but sometimes it's just what you have, and that's better than nothing. And I think there's a healthy balance between just going on the results of 2 patients. That might be not enough, but there's a balance, I guess.
Well,
and
showing proof of concept, I think, in and of itself is a very important thing to accomplish from a research innovation standpoint. So it shouldn't hold you back. I was just curious how you handled that.
Yes.
Thank you.
No, this is the way we handled it. We were aware, but it was everything we could do.
Thank you. Thank you for popping on there really quickly to help to provide some additional context. So thank you. Next question. What do you think would be the ideal way to fill the EHR? More open fields or structured fields?
Oh, that's very interesting. That's also something we're working on now at the ministry, of course. I think there will be more structured fields, but in a way it's still accessible for healthcare professionals, of course. If you're just filling in forms, no one will be happy. So it needs to be a smart structured field. And I'm always curious how stuff as AI, well, you cannot just implement it, but can indeed get context from the open field into a structured field. So I'm hoping in the future that will be also a part of it. But for now, at least structuring in a way that everyone structures in the same way. I think that's the most important we need to do. And then it might not even be, yes, and it might not even be more or less structured, but if we structure in the same way, that would be nice.
Yeah. I know if everyone did just think about it in terms of what we need for research, that'd be excellent. However, when it comes to actual clinical practice, yeah. A couple more questions and then we'll wrap up. Next one comes from Pim. How can informed researchers in the Netherlands to make the aware of SNOMED CT and researchers in other countries?
Well, I just found it, I was aware of it in some articles I bumped into, but I think, yeah, that's somehow still incorporation in research articles. That's the best way, I think, how to inform researchers. That's where they're looking for information. And maybe this is just on top of my head, but the combination of where you publish or where studies with SNOMED are published, if they are in a certain field, and how do you attract the certain researchers you want to know about it.
Mhm.
If you only publish in very informatics related journals, but you want to show it to the physicians, well, they might not read it. And I think there's or it might not be, they might read it, but it's completely not understandable. So it might be something around that. And it's always difficult, I guess.
Excellent examples. I hail from neuroscience and also a library background, and one area that heavily focused on for a number of years is embedded librarianship. And so where the librarians would be right there with the researchers. And one aspect of that was actually presenting at their conferences and not always at the librarian conferences. So it sounds like it's very similar to what you're expressing there. SNOMED CT, SNOMED International, our SNOMED community, definitely, going out to the areas of research that are specific and not just our SNOMED or our heavily informatics events, I think would be highly beneficial, as you say.
And I think you might actually surprise people that it exists, and people would be like, "Oh, this is actually nice. How can I use it?" And then just continue.
Like you said, you've been involved in biomedical research, and it wasn't until last year or so that you've really become heavily involved. I mean, attending SNOMED CT conferences and everything this year, and so it's been interesting to see even your evolution in the use of the clinical terminology standard.
Yes, exactly.
Last question here. Do you have any ideas about how to increase structured data entry? Is the main cause of the 80% data being unstructured users not finding suitable matches for their entities, or by software limitations, or by lack of training?
Oh, there's a lot of layers in that question.
Yeah.
Well, one thing I didn't research, of course, if the 80% was also accurate for my health records. So that to start with, so it's a number that just comes up in papers, and at a certain point it has its own like truth in it. How to increase? Well, that's also a question I'm trying to answer at the ministry. It would be nice if I would have known the answer. But I guess it's a lot of different layers. So you need to be in contact with the physicians, the people, how to fill in those records. They need to be trained. They need to know the relevance, and they also need to get it back. So if there's no relevance for them to do it in that way, of course they're not going to do it. But also the records need to be in a way that it's easy. And I think it's a balance between those. But that's nicely said, and it's very difficult to achieve just in a second.
Well
So
said. That was a loaded, very high level theoretical. Okay, so we are at the top of the hour, and so I just want to kind of wrap things up. As I mentioned, the slides and the presentation will be posted later this week or early next week, and I'll be informing everyone who has registered of when those are posted and how to access those. Until next time, I hope to see you all at the next SNOMED CT research webinar. Also look for the announcements for the registration for the SNOMED CT Expo happening in Atlanta, Georgia, United States in October. Sylvia, thank you so much for this excellent presentation. That was an exciting lot of research, a lot of work that you've been involved in, and that was absolutely exciting. And other than that, I will see you all next time. Thank you so much for attending.
Yes, thank you all.